Hello and welcome to our mini lecture on DNA replication and mitosis. Our DNA, or deoxyribonucleic acid, is housed in the nucleus of the cell. The purpose of DNA is to store, transmit, and express genetic information that determines how living organisms grow, develop, function, and reproduce. Our DNA is made up of genes, which contain the instructions for the creation of proteins within our bodies. Watch our video on protein synthesis for more information on that. In order for our cells to divide, our DNA needs to be replicated so each of the daughter cells can contain an entire set of our DNA. Let's break down how DNA is stored in our cells. DNA is made up of two strands of repeating units of nucleotides. Each of these nucleotides has a nitrogenous base. There are 4 different nitrogenous bases within DNA: adenine, thymine, cytosine, and guanine. And they're indicated, which you can see down here, by the first letter of their name. So A is adenine, T is thymine, C is cytosine, and G is guanine. The 2 strands of DNA are held together by hydrogen bonds. You can watch our video on atoms and bonding for more information about hydrogen bonds. The bases of one strand of DNA always pair with the base of the other strand of DNA in a very specific way called complementary base pairing. Adenine always pairs with thymine using 2 hydrogen bonds, and you can see that on the left here. Cytosine always pairs with guanine using 3 hydrogen bonds, which you can see here. So A and T go together, C and G go together, and this is always the case in DNA. These base pairs then form the rungs of this DNA ladder. If we were to connect all of these, you can see how that makes a ladder there. The 2 strands of DNA then twist around one another to form this double helix shape. So that's the term for what this shape here looks like. The double helix then wraps around proteins called histones, which you can see up here. This DNA that's loosely coiled around the histones is called chromatin. And this is how DNA is stored in a non-dividing cell, so a cell that's at rest. When a cell prepares to divide, the DNA condenses further into chromosomes. In order for a cell to divide through mitosis, it must first make a complete copy of its DNA. The DNA replication occurs in 3 phases. Phase 1 is called initiation. During this phase, the 2 strands of DNA are separated by the enzyme helicase, which you can see here separating that double helix of this DNA. Stage 2 is called elongation. During elongation, each of the original DNA strands becomes a template strand to build the new complementary strand of DNA. So you can see this is the original strand of DNA, and this is this new template strand that's being built underneath it. The enzyme DNA polymerase is what brings the complementary nucleotides to the template strand to create that new strand. Stage 3 is termination. Once the 2 original strands are both bound to their newly created complementary strands, the cell is left with 2 complete sets of DNA, and replication terminates. These 2 sets of DNA each contain 1 original strand and 1 newly built complementary strand. Additional enzymes then scan the newly created DNA strands to correct for any potential mistakes that may have been made during replication. Most of the cells in our body divide regularly through the process called mitosis. These cells are called somatic cells, and they include all of the cells in our body except for germ cells, which are cells that produce eggs and sperm. Somatic cells have 2 copies of each chromosome, 1 from each parent. Human somatic cells have 23 sets of chromosomes, which make them diploid cells. In contrast, germ cells divide through meiosis and produce haploid cells, each containing 23 single chromosomes. You will learn more about meiosis when learning about the reproductive system. The cell cycle refers to the life cycle of a cell. It begins when a cell is created through the division of a prior cell and ends when a cell divides to form 2 daughter cells. The cell goes through multiple phases during its life cycle. including interphase, mitosis, and cytokinesis. The cell spends most of its life in interphase, which is further divided into multiple stages. So you can see here that this little M phase, that's mitosis. The rest of the cell cycle is all considered interphase. Cells that are not actively dividing are considered to be in the G0 phase, which you can see over here. The G0 phase is the resting state where the cell performs all of its normal cell functions and is not actively working to divide. When a cell gets ready to divide, it first enters stage G1, which you can see here. The G stands for gap, so G1 is gap phase 1, or the growth phase of the cell. The S phase follows the G1 phase, which you can see here. DNA replication takes place during the S phase in preparation for cell division. The S stands for synthesis, as the cell is synthesizing new DNA. The third phase is G2, or gap phase 2. This is the second growth phase of the cell, where it prepares for mitosis. It should be noted that the G1 phase can last hours to days, whereas the S phase lasts about 8 to 10 hours, and the G2 phase lasts about 5 hours. After the S phase of DNA replication, the cells contain two copies of DNA in the form of sister chromatids that look like an X and are connected at the centromere, which you can see here, this yellow is the centromere. So in a non-dividing cell, we have these two homologous chromosomes. In the dividing cell, once we have replicated our DNA, we now have two copies of each of those strands of DNA, one from each parent. During mitosis, you will see that the sister chromatids are pulled apart, so each resulting cell ends up with a single copy of the DNA. Mitosis is the phase of the cell cycle where the cell is actively dividing. It's followed by cytokinesis, where the single cell is cleaved into two separate cells. Let's take a look at each of these phases of mitosis. We'll start here on the left with prophase, which is the first phase. In this phase, the chromosomes condense and become visible in the nucleus, and the nucleolus disappears. So we've gone from chromatin, where the DNA is loosely coiled around those histones, to now they've been condensed into chromosomes. The spindle fibers also emerge from the centrosomes, and the centrosomes move toward opposite poles of the cell. So you can see these centrosomes here moving toward opposite poles of the cell. The last thing that happens is the nuclear envelope begins to break down. Our next phase is prometaphase, where the chromosomes continue to condense. The kinetochore, which serves as the attachment site for the spindle fibers, appear at the centromeres, and the spindles attach to the kinetochore. So these spindles you can see here in blue, and they have attached to the kinetochore at either end of the cell. During metaphase, the chromosomes line up at the metaphase plate in the middle of the cell. So when you see the chromosomes all lined up like that, that definitely shows you that you are in metaphase. The spindle fibers then attach to each of the sister chromatids at that metaphase plate, which is the center of the cell. The next phase is anaphase. During anaphase, the centromeres split apart and the sister chromatids are pulled toward opposite poles of the cell. The cell also begins to elongate. So you can see how it went from a sort of circular in prophase to now it's beginning to be more of a football shape in anaphase. The last phase of mitosis is telophase. During this phase, the separated chromosomes begin to unwind and a new nuclear envelope appears around each set of chromosomes. The mitotic spindles break down and the spindle fibers continue to elongate the cell. So now we have a nucleus on either side of the cell. Following mitosis, the cell contains two separate nuclei and it needs to be divided into the two daughter cells. This occurs during cytokinesis. In animal cells, the process starts with a cleavage furrow, which eventually separates the cell into the two distinct daughter cells. Thank you for watching our video on DNA replication and mitosis. Stay tuned for our next video about protein synthesis. We'll see you next time.